So hello class and welcome to week 9 of the next generation sequencing analysis course. We begin this week sort of at a juncture in the class rather, where we have been through a lot of the methodologies in whole genome resequencing, and beginning next week we'll be shifting gears and heading into the world of RNA-seq and gene expression analysis with short-read data. So what I'd like to do this week is to kind of tell you about some of the challenges that we face in the analysis of the data once we have conducted some of the preliminary sort of processing steps that we've talked about. And we encounter a lot of issues with data management with these very big data sets, and so I'd like to tell you about some of the tools that we use and some of the challenges that we face. So I'd like to begin though, because your homework is related to genome intervals, and I'm going to try to convince you that essentially all of the data that we collect and the features that we analyze in whole genome data can essentially be thought of as sets of intervals. And so when we're talking about intervals, we need to be clear on our notation and our formatting. And we also, because we have so many intervals in our analyses, including thinking of a short read mapped to an alignment, we can think of as an interval because it has any short read has a 5' and a 3' coordinate, and therefore we can call it an interval. So we have a lot of intervals because in a common short read data set we might have 300 or 400 million aligned reads, together with all of the features in the genome that we can also think of as intervals, such as an exon, which has a defined 5' and 3' position in a reference genome. So there's a lot of things that we're interested with respect to intervals, and we need to be clear on our format. So BED is a feature format meant to directly deal with specification of intervals. And the BED is called the Browser Extensible Data Format. It'll become clearer why we call it the Browser Extensible Data Format in a moment. But here at the top, what we have is just a simple set of coordinates on chromosome 7 in what we call BED6 format. So just notice here that in the first column we have a chromosome, then we have two 5' and 3' coordinates, together with some metadata here that I'll describe in a moment. But the important thing to know about BED format is that the first coordinate is what we call a zero-based coordinate. So BED is a zero-based coordinate system. So what does that mean? Well, what it means is that this coordinate here does not actually refer to that position. It refers to the position one base pair downstream, or towards the 3' end. So this position here is actually 127 megabases, 471,197 is the position that this coordinate actually refers to, not 96. So this is a zero-based coordinate system because it begins counting at 0, not at 1. So we must add 1 to this coordinate in order to obtain the base position in the reference genome that we're referring to. Okay, so let's take an example, a little smaller number here. Imagine chromosome 1, position 100 to 200. In a related format, GFF, that coordinate position on the left here would be referred to as position 101. So this is a zero-based start coordinate system, but a one-based end coordinate system. So notice here that GFF is essentially the numbers that we're used to working with. When I refer to position 101, I mean count up 101 positions from the beginning of a sequence, and that's the position I mean. And 200 likewise. So just again, BED format, zero-based begin, one-based end. GFF is one-based begin, one-based end. So why use zero-based coordinates, you're probably wondering? Well, it all has to do with how we calculate the length of a coordinate, or an interval rather. So for SAM and BAM, VCF, GFF, most of our coordinate systems that we talk about, to get the length, you simply take the end coordinate minus the beginning coordinate, but you need to remember to add 1. This is essential. So let's take an example. Imagine we have an interval between 1 and 10. To get the length of that interval, we all know that the length is 10 from intuition, but to get the length of that interval, we must take the end coordinate 10 minus 1, which gives us 9, but we must add 1 more to get the length of 10. Now notice for a zero-based coordinate system where that might be, that same interval would be reflected as 0 to 10, not 1 to 10. If we take the end of 10 and subtract the beginning of 0, we get the length of 10. But notice that we didn't have to do an additional mathematical operation to add 1. This is the point of browser extensible data format, okay? Because when you're working with so many intervals, as we are with short read data, when large computer servers, particularly web-based servers where individuals are trying to visualize big data, these calculations begin to impose a computational burden, hence the reason for only needing to do 1 versus 2 computations. Okay, but that's the essence of it. Now I wanted to introduce some notation here because you commonly see these notations, and for a long time I was felt they were a bit cryptic and didn't totally understand them myself up until I started to deal with the bed format extensively. But here's sort of just the essence of it here. An open interval means we're not including the first position. We must first add 1 to obtain the first position in the reference genome. So this means that first position is open. So if this is a 0, we're not actually referring to position 0, we're referring to position 1. Okay, so we had to add 1 to that first position. So it's actually open, what we call zero-based half open, because the end position is closed, or 1-based. Okay, and this is the notation for open and closed. So all of this is a bit cryptic. You'll see this in various discussions and people discussing different types of intervals, and so it's just useful to know what we mean when we say open and closed. So bed is also sort of extended to multiple different columns. It doesn't just involve the start and end positions of a coordinate or an interval rather. The first column of bed formatted tables always contains a chromosome contig or scaffold name. The first position is that 0-based coordinate. The end is that 1-based coordinate. Then we have a feature name, some score or value. This could be even text. So these are some examples, like a p-value. So if we had an interval that was an mRNA and we were interested in whether or not it was differentially expressed, the score here could be a p-value. We could have an enrichment score for a feature to say some PFAM domain is enriched in some protein expression data set. That could be there. We could even incorporate text in the score, etc. Finally, for bed 6 anyways, we can have the strand of the feature. So for some applications, we need to keep track of the strand, such as an mRNA, which is complementary, which reflects the sequence of one strand and not the complementary strand. So I'd like to refer to you to some of the bed tools documentation, which describes the essence of some of this perhaps more eloquently than I can. I'd also like to refer you to the UCSC, the University of California Santa Cruz site, for a description of many of the different formats that we'll be talking about and have talked about. This is kind of a commonly, or a well-frequented website for looking up some basic descriptions of common formats in next generation sequence data. So I mentioned bed tools here. I haven't actually introduced that to you other than in the week 9 homework, where you conduct a series of exercises using bed tools. As I mentioned in the homework, the documentation at the bed tools site is perhaps some of the most superior documentation of any software for bioinformatics analysis, and so there's a lot of goodies there in terms of grasping some of the concepts involved in interval data and how to go about doing interval-related analysis. So there are additional bed formats, that include, bed 7, bed 8, all the way to bed 12. That's just referring to the number of columns. These are all tab-delimited columns, by the way. It's just a simple flat file type table format with sort of rigorous definitions of the individual columns. So what do we mean by intervals? Again, any aligned data type or genome feature that has genomic coordinates we call an interval. So short reads from our Illumina data or our solid or our 454 data can be thought of as intervals simply because they can be mapped and therefore have 5' and 3' coordinates. Genomic features such as exons, introns, SNPs even. SNPs are in fact have intervals. In bed format, a SNP at position, say, 3 in the genome, you guessed it, its 5' coordinate in the bed format is 2, and its 3' coordinate is 3. Okay, because again, bed is 0 base, so even a feature of length 1, in bed format has the interval 2 to 3. Of course, in GFF or in VCF, that the intervals are, of course, the 5' end 3 and the 3' end would be coordinate of 3. So all of these can be thought of as intervals, and because they can be thought of as intervals, we need to just reconcile that we oftentimes are interested in comparing intervals. So imagine we wanted to determine which features in a genome overlap. So here we have one set of intervals in blue. Okay, that could be, say, repetitive elements, and B here could be a separate set of elements, say genes, for example. And so we have a set of repetitive DNA, and we have a set of intervals that our gene prediction software considers genes, and oftentimes we're interested in the question of which features overlap. Okay, so we can answer that question here in green, which shows the intervals, the positions, the 5' and 3' coordinates of the overlaps between these two sets of data. So this is a common question: which features overlap? Of course, we can also ask how many features overlap. Okay, in this case, 2 features from the A set of intervals, or our repetitive DNA, and 4 sets of 4 intervals in our B set overlap with that set from A. We could also ask how many base pairs do they overlap. Okay, here we're simply interested in counting the number of base pairs in the overlapping intervals. What is the DNA sequence in the overlapping intervals? Another common questions. And perhaps which features don't overlap. So this, of course, would be a feature in the set of repetitive intervals that don't overlap. So Bed Tools is really the premier software for analyzing this type of data. There are other softwares available. You may someday encounter a type of software called Kent Tools, which has some early attempts at calculating interval data. There's also an R-based system for working with intervals that is called, the R-based system is called G-range or G-range objects. These are another way in which in the R statistical language that there's a system in place for conducting analysis of genome intervals. So why bed tools, though? This is the one that I want to introduce to you, and the one that I favor myself for a variety of reasons. So as I've described, we're dealing with massive sets of intervals with our short read data. It becomes computationally challenging. So if we wanted to identify, say, all of the features that overlap with our short read data, so all of the, say, features in a GFF, all of the introns, exons, gene features, etc., that are in our function, in our structural annotation, we want to know which of those overlap with our short read data. Maybe our short read data is ChIP-seq data, where we fished out regions of the genome that bind to a particular protein. Maybe we then want to, fish out all of the, transcription known transcription binding sites that match our ChIP-seq data. What we're talking about is enormous computation, and there are some serious challenges to that. And BedTools has come up with a simple way of doing those calculations for us. So another advantage is that BedTools has a simple command line interface. We also BedTools will also read sort of commands in from standard in and will output to standard out. So as you guys are now well versed in sort of Unix command line programs, you'll be comforted to know that you can pipe BedTools commands to additional BedTools commands or to Unix commands such as grep, etc. So this all provides a really flexible set of tools for answering the questions that we have about these interval data. Okay, so because you'll be working with this extensively in your homework, let's just go through a few examples, because I don't want this to sound too cryptic. Actually, this is the essence of much of sort of next generation sequencing analysis is going about summarizing interval data. So here's some examples. So intersect bed. So I want to make one thing clear, that BedTools went through a transition where in nomenclature for their binaries. So intersect bed here is the old command, okay, for conducting the most basic analysis of which positions in a set of intervals, so set A and set B, like in the cartoon a couple of slides back, which of those intervals overlap each other. And intersect bed is the main binary to answer that question, but the nomenclature has changed so that it's no longer intersect capital B bed. It is simply now we type BedTools intersect.